Hello and welcome. My name is Gina Suh, and I'm an infectious disease physician leading our phage therapy program here at Mayo. I would like to welcome you to our Advancing Care Through Genomics virtual speaker series focused on phage therapy. Today is the third in our multi-part series. Dr. Nicholas Chia is joining me today as co-moderator for the event. Dr. Chia is an associate professor in the departments of surgery and laboratory medicine and pathology, and co-director of the microbiome program here in our Center for Individualized Medicine. Before I introduce our speaker, a few housekeeping items. Please use the Q&A in Zoom to submit your questions, which we will take at the end of the session. For those interested in earning CME credit, an email will be sent within 48 hours that includes instructions on how to claim credit. We are delighted to welcome Dr. Julia Willett as our featured speaker. Dr. Willett is an American Heart Association postdoctoral fellow in the laboratory of Dr. Gary Dunney in the Department of Microbiology and Immunology at the University of Minnesota. She uses genetic and biochemical tools to understand the molecular underpinnings of bacterial biofilms and interbacterial interactions using Enterococcus faecalis as a model organism. Her work focuses on determining the function of poorly characterized gene products that contribute to biofilm development or mediate bacterial interactions. She is especially interested in understanding how E. faecalis responds to antibiotics and how antibiotic-resistant biofilm-associated infections may be targeted by phage therapy. Recently, Dr. Willett published the work on the first type 7 secretion system, or T7SS, characterized in Enterococcus. When induced by phage or antibiotics, the E. faecalis T7SS can restrict the growth of neighboring gram-positive bacteria. Dr. Willett is also passionate about developing genetic tools such as arrayed transposon libraries that enable high-throughput genetic screens in understudied pathogens. Her postdoctoral research was previously supported by an NIH/NHLBI T32 training grant. Prior to her postdoctoral work, Dr. Willett earned her PhD in molecular, cellular, and development biology at the University of California, Santa Barbara, where she studied contact-dependent growth inhibition systems in gram-negative bacteria. As a graduate student, she co-authored 7 scientific publications and was supported by an NSF graduate research fellowship. Dr. Willett's presentation today is entitled Phage and Antibiotics, Induction of an Antibacterial Type 7 Secretion System in Enterococcus faecalis. Welcome, Dr. Willett.
Thank you so much for that really lovely introduction. I'm really excited to be here. There we go. Yeah, today I'll be talking about phages and phage therapy and how it relates to the model organism that I've studied primarily during my postdoc, which is Enterococcus faecalis. So the background image on this slide is a nice image of an Enterococcus biofilm, which is one of the things that we study a lot in our lab. So just as a brief overview of what I'll talk about today, I'll start by introducing Enterococcus as a commensal and as a pathogen. I'll talk about some work that we know about Enterococcus phages and how that relates to phage therapy. And then I'll tell you a really interesting story that arose out of a collaboration between our lab and Breck Durkop's lab at UC Denver, which looks at how Enterococcus responds to phage infection in vitro and some consequences that this might have on neighboring bacteria. And then I'll wrap up with some conclusions and some of what I think are kind of the driving questions in the field for the future. So Enterococcus faecalis is a ubiquitous commensal organism that colonizes between 60 to 80% of infants within the first week after birth. It grows to high levels after this initial colonization, but then the levels drop once the microbiome starts to mature. However, it sticks around at low levels in most people and is found in about 80% of the GI tract of healthy adults. And it can also be part of the normal healthy microflora that's found in the oral cavity. Enterococcus has several probiotic properties, and the strain Symbioflor-1 is a safe, commercially available probiotic, and some other strains have been used as starter cultures for fermented dairy products. However, it has this sort of dual lifestyle wherein it can transition from a commensal organism to a really deadly opportunistic pathogen. Enterococcus can be highly antibiotic resistant through through intrinsic and acquired antibiotic resistance mechanisms. And so after antibiotic treatment, Enterococcus can overgrow in the gut, which can facilitate this transition or this translocation out of the gut into other areas of the body where it can act as a pathogen. Enterococcus can also produce a number of protein factors that helps promote this translocation outside of the gut. And also in the oral cavity, Enterococcus can persist in chronic infections even after antibiotic treatment. So it can be a real problem for things like infected root canals in the oral cavity. And there are several properties that make Enterococcus faecalis an effective commensal organism and also an effective pathogen. So it can attach directly to host tissues and form biofilms, which helps it stick around as a commensal in the gut, but also leads to chronic and persistent infections that are difficult to remove or eradicate. Colonization also doesn't seem to provoke a strong immune response. And so then when it transitions to a pathogenic lifestyle, it can kind of evade the immune system, which also is associated with some chronic infections. And one other important thing to note is that Enterococcus faecalis can also be spread via contaminated surfaces, as Enterococcus faecalis is really resistant to things like heat and desiccation and chemical cleaners and other things that we might use to disinfect surfaces. So when Enterococcus does get outside the gut, it can form infections in many areas, including infective endocarditis and infections of driveline material attached to LVAD devices. It can form prosthetic joint infections and infect things like chronic wounds and surgical sites. And I think it's really important to highlight that many of these infections can be polymicrobial, which can increase the severity and the persistence of the infection. And one other thing that contributes to Enterococcus infections is biofilm formation. So Enterococcus is really good at forming biofilms, and what I'm showing here on the top is a cartoon diagram of sort of typical biofilm formation. So biofilms start to form when planktonic or free-floating cells find and attach to a surface. They then develop into or start producing an early biofilm surrounded by a matrix made up of things like extracellular DNA and polysaccharides. And they ultimately mature into a mature biofilm. And in some cases, we know that bacteria can leave or break off the biofilm through a dispersal process, returning to a planktonic state where they can then find and attach to a new surface and produce a new biofilm. And we've done a lot of work looking at biofilms in vitro and in vivo, and we know that in vitro and in vivo, Enterococcus faecalis forms fairly similar biofilms. So in vitro, I'm showing a cartoon here of what in vitro biofilms kind of look like. They're mostly these flat monolayers, sometimes with cells piled on top of each other, and they're surrounded by an extracellular matrix. And we can visualize that here. This is an Enterococcus biofilm grown on a polycarbonate coupon from a paper our lab published a few years ago in collaboration with the Mayo Clinic. And here you can see individual cells and that extracellular matrix material stretching between the cells. And we also have done a fair amount of work, again, in collaboration with the Mayo Clinic, looking at biofilm formation in the GI tracts of germ-free mice. And so we know in vivo, Enterococcus forms these small clusters that we call microcolonies, where there's just a few layers of cells piled on top of each other, encapsulated in a matrix. And here's a micrograph of what that looks like on the epithelial surface of a germ-free mouse gut. So, like I mentioned earlier, many Enterococcus infections can be polymicrobial, and we know that there are some specific interactions that promote these infections with other bacteria, or can actually, Enterococcus can actually antagonize other organisms. So we know that some strains of Enterococcus produce a compound called EntV, which can inhibit hyphal morphogenesis of Candida albicans and prevent biofilm formation and virulence of this organism. There are many strains of Enterococcus that produce bacteriocins that can antagonize other organisms. However, Enterococcus can also promote polymicrobial infections, in some cases by decreasing the immune response to other pathogens, and in other cases by promoting the growth of polymicrobial biofilms, which are very difficult to treat. And in some cases, we know that certain strains of Enterococcus can sort of confer antibiotic resistance onto other organisms or produce metabolites that promote the growth of these other organisms. And again, all of this kind of contributes to the difficulty that arises in treating Enterococcus infections. So I now want to just share some of the information that we know about phages and phage therapy in Enterococcus. In terms of the study of phages and phage therapy, Enterococcus is a little understudied compared to some other organisms. And so briefly as a review, phages are viruses that infect bacteria. They can have either lysogenic or lytic cycles, and lytic cycles where the phage replicates and bursts out of the bacteria, killing the bacterial host. These are the phages that we think of as suitable for phage therapy. And again, phage therapy is just the application of phages to treat bacterial infections. So a couple days ago, I used this lab website to see how many phage genomes of phages that target Enterococcus were available. And I was able to find 61 unique Enterococcus phage genomes. So these are genomes that have been deposited through NCBI. So again, there is a lot of really good work out there on phages that target Enterococcus, but we still don't have quite the number of phages isolated against this organism as we do for some other organisms. And I think it's really interesting to note that many Enterococcus phage studies have been done using phage isolates from the oral cavity. So there's been a lot of work in the field of oromicrobiology, isolating phages and then using in vitro models to model the treatment of chronic infections in the oral cavity using phage therapy. But there hasn't been quite as much work done developing models for sort of other Enterococcus phage infections or looking at phage therapy, say, in the GI tract. We also know that of the Enterococcus phages that are available, not all of them would be suitable for phage therapy. So in Enterococcus phage genomes, there are some that encode beta-lactamases that are related to gene products that mediate antibiotic resistance. We know that phages can transfer antibiotic resistance between Enterococcus species in some cases. But some work that's been done in the last few years has isolated some obligatelytic phages, including this VPE25, which I'll talk about later, and that these might be better candidates for phage therapy. And one other thing that is sort of interesting about Enterococcus and phages is that there have been several studies that have shown that many Enterococcus isolates have a lot of lysogens or prophages. I think the strain V583 has 7 prophages, which is about 10% of its genome. And we know that excision of some of these is inducible by compounds that damage DNA, UV light, but we don't really know how induction of phages might affect an infection if there's an isolate that has some of these lysogens or prophages, or the effect that this might have on sort of other bacteria. So I briefly want to highlight some of the in vivo approaches that have been done looking at suitable approaches for phage therapy of Enterococcus faecalis. And this table is from a review paper last year. And I first want to draw your attention to the red box up at the top of the table. And this is currently the only known study on phage therapy for Enterococcus infections that's been done in humans. And it was a small cohort of patients from Poland that had chronic prostate infections. So they received application of a phage lysate. And in all 3 cases, the phages targeting Enterococcus faecalis gave encouraging results, including a reduction of the bacterial load and a lack of early disease recurrence. But there isn't really anything else that's been done in humans at this point. So the other lines on this table here summarize some of the animal models that are available for Enterococcus phage therapy studies, and some of the work that's been done in those looking at infections like bacteremia and sepsis. And one thing that I find interesting about this is that these studies have used a really wide range of doses of phage, so all the way down from 10 to the 3 or 10 to the 4 PFU per mouse, all the way up to 10 to the 12th PFU per mil. So several orders of magnitude difference in the phage applications that have been tried. And there's also been some work done using specific purified endolysins or lysins that are found encoded by phage to see if these would be effective at eradicating infections instead of using the complete phage particles. And in many cases of these studies, phage therapy was associated with a decrease of disease severity, an increase of survival in the animals. And there were a couple of studies that looked at the effect of this phage therapy on the microbiome, and they didn't find any major differences in the microbiome of animals that had received phage therapy, indicating that specifically using phage to target Enterococcus faecalis wasn't associated with really wide dysbiosis of the rest of the microbiome. There was also a study from last year that found that alcoholic hepatitis in a cohort of patients, mortality from this disease was correlated with Enterococcus faecalis strains producing cytolacin toxins. And so the researchers from this paper, they took fecal samples from these patients. These fecal samples contained the Enterococcus strains producing these toxins. They did fecal transplants into mice to humanize their GI tracts, and then they targeted these Enterococcus strains with phage. And what they found was that in these mice, the mice that received phages targeting these Enterococcus strains had reduced liver damage, suggesting that phage therapy might be an effective way to decrease the disease severity of human patients with this disease. So I think there's been a lot of great progress in terms of animal models and ways that we can sort of go forward with figuring out the best approaches for Enterococcus phage therapy. I next briefly want to highlight some work that's been done in vitro, specifically looking at the effects that phage therapy might have on Enterococcus biofilms. And again, this is super important because, as I mentioned earlier, so many infections caused by Enterococcus are biofilm-associated, and we know biofilms are much harder to break up than liquid planktonic cells. And so I think it's really important to consider sort of biofilm models when we're looking into developing phage therapy approaches against Enterococcus. So this first study from 2019 used a phage cocktail, and they applied it to biofilms that were grown in this in vitro collagen wound model, where they take collagen and coat the surface of plastic dishes to kind of mimic a more in vivo environment. And what they found was that the phage treatment was associated with a significant reduction of viable cells from these dual species biofilms formed between Enterococcus faecalis and Enterococcus faecium. And interestingly, they found sort of the best reduction in the level of viable cells at early time points compared to a full 24 hours after phage treatment, suggesting that this type of targeted phage therapy might have some initial success early, but that cells in a biofilm might be able to recover at later time points. Another study from 2019 used a different phage cocktail, the phage cocktail by itself or a phage cocktail embedded in a polymer mixture designed to prolong the release of these phages. And they applied this to V583 biofilms grown in plastic dishes. And V583 is a vancomycin-resistant isolate of Enterococcus faecalis. And what they found was that either the cocktail or the polymer mixture had about a 90% reduction in viable cells and from these biofilms. And they actually carried this study all the way out to 4 weeks, which I thought was great, really kind of looking at these sustained applications of phage therapy. And they found that this cocktail and the phage polymer mixture still had a good reduction of viable cells after several weeks of treatment. And finally, there is another study from a few years ago, again from the oral biology field, where they used this engineered phage. I believe this phage was previously a lysogenic phage, and they've engineered it to become lytic. And they used an in vitro model for infected dentin root segments, where they can inoculate these root segments with Enterococcus strains and the phage, and then quantify the reduction in viable cells. And what I found striking about this study was that for B583, this vancomycin resistant strain, they found a 99% reduction in viable cells in this in vitro Denton model. But when they looked at another strain, strain JH2-2, they found only about a 20% reduction of viable cells. So this is really interesting to me because I think it really highlights that, when we're considering phage mixtures and phage cocktails and using it to target different strains of Enterococcus, we need to be mindful of really big strain differences that might arise in these systems. So I next want to highlight some work that's been done looking at the interactions between phage resistance and antibiotic sensitivity in Enterococcus. And there have been a couple really great studies showing that an increase in phage resistance might actually lead to a decrease in antibiotic resistance. And so in this paper or the figure that I'm showing here on the left, the OG1RF Enterococcus faecalis strain is sensitive to phage, and then they're evaluating the MIC of daptomycin here. And in this, these two strains marked by red arrows, each of these strains are phage resistant. This one, I believe, was an evolved isolate in the lab. And this one is lacking a gene that's part of the extracellular polysaccharide that the strain of Enterococcus produces. And in both cases, these phage-resistant strains have a lower MIC to daptomycin, suggesting that they're more susceptible to this drug. And a similar thing was shown with vancomycin, where this strain here shown in red is a strain that's resistant to phage. And it is more susceptible to vancomycin than the wild type version of this strain. And some work from our own lab a few years ago, we had an ID fellow who was interested in studying this relationship between antibiotics and phage. And she was using a model where she could grow biofilms on the driveline material used for LVAD devices. And so she would grow biofilms on these sections of driveline and then treat with either a lytic phage NPV1 or with ampicillin or with a cocktail of the phage plus ampicillin. And then when she quantified surviving cells, what she found was that the phage plus antibiotic cocktail was more effective at removing cells from the driveline and decreasing the number of viable cells than either treatment alone. And so I think this really highlights that there's some potential for really studying, this relationship between phage resistance and antibiotic sensitivity, and hopefully it can lead us to develop some really great treatments of either, combining different phages or combining phage and antibiotics. Okay, so I next want to talk about some work that I've done in collaboration with Breck Durkop's lab at UC Denver. And these experiments have been focused on understanding how Enterococcus might respond to phage treatment and how that might impact the surrounding microbial community. This work was just accepted for publication in PLOS Genetics. And if you're interested, the preprint for this publication is available on bioRxiv. So the phage that we'll be talking about for the rest of the work is VPE25, which is shown here. And this is a phage that Breck isolated from sewage in Dallas during his postdoc. It's a lytic phage that has activity against the strain OG1RF of E. faecalis. And so I'm showing that here in this figure where in the absence of phage, this strain can grow fine on agar plates, and in the presence of the phage, growth is prohibited. Breck mapped the specific protein receptors required for target cell recognition by this phage, and he and his postdoc Anushala showed that mutations in the polysaccharide that's produced on the surface of Enterococcus faecalis is also associated with resistance to phage. And so that's what these strains here are showing, or that if you mutate the receptor required for phage, or if you mutate the genes that are required to produce polysaccharide, you get resistance to this phage in vitro. So OG1RF, the strain that we're using, is a plasmid-free strain that we use in lab. It's susceptible to this phage again. And it also forms robust biofilms and is a derivative of a clinical isolate originally obtained from dental caries back in the 1970s. So we think that it's a really good model, model strain that we can work with easily in lab. So Breck and Anushla wanted to know how OG1RF responds to phage treatment, or what gene expression changes happen after it's been treated with VPE25. So they infected OG1RF with this phage, then harvested RNA at 3 different time points for RNA-seq experiments. So what this heat map is showing is a summary of the changes in gene expression at multiple time points after infection with this phage. And what they found is that there are specific gene expression changes or specific patterns in gene expression changes that happen at early, middle, and late time points after infection. So 10 minutes, 20 minutes, or 40 minutes after treatment with this phage. So there are distinct temporal changes that the cell goes through after phage treatment. And they also did some transposon sequencing experiments with a transposon library that we have in our lab. And they identified specific mutations associated with phage resistance, some of which I showed on the previous slide. So I got really interested in this project after seeing part of their RNA-seq results. And so this volcano plot shows changes in gene expression 40 minutes after phage infection. So this is kind of the late time point that they used. And I want to draw your attention to these circled genes down here in this yellow box, which show genes that encode a type 7 secretion system. My research background is in bacterial protein secretion systems, and my graduate work was on secretion systems in gram-negative bacteria and how they mediate bacterial competition. So I became really excited to look into this and see whether this secretion system could mediate bacterial competition in Enterococcus faecalis. So what are type 7 secretion systems? Type 7 secretion systems are protein complexes in bacteria. They're found throughout Firmicutes, and they're related to the type 7 secretion systems or the ESX secretion systems that are found in mycobacteria. And we know that they're important, those ESX systems are important for virulence in mycobacteria. There are several core membrane proteins that form this type 7 complex and work to secrete effector proteins out of the cell. And so in the figure here on the left, this is a diagram of some of the sort of the core conserved membrane proteins that make up the type 7 secretion system machinery. Some of these proteins recognize other proteins that are called effector proteins, and they export them out of the cell in an ATP-dependent fashion. They can deliver these effector proteins into neighboring bacteria in a contact-dependent manner. So it requires contact between the cells. And then these effector proteins can be delivered either to the cytoplasm or possibly to the cell wall of these recipient bacteria. So there are essentially 2 classes of type 7 secretion system effector proteins. And several of the ones described in either Staphylococcus or Streptococcus are toxins that kill other bacteria. So the first set of effector proteins are these so-called WXG100 effectors. They have this name because they're about 100 residues long, and they have a WXG amino acid motif kind of in the middle of the protein. And in tuberculosis, these proteins are also called CFP10 and ESAT6, and these are some that are associated with virulence of this organism. Other effectors include the LXG-domain proteins, and they're called this because the end terminus of these proteins encodes something called an LXG-domain. And several of these, like I mentioned, are actually toxins that can kill other cells. So in Staph aureus, there's an LXG protein called ESAD that's an endonuclease, so it can cut nucleic acids like DNA and RNA. There's a really nice paper published earlier this year showing the discovery of a protein called TSPA, also in Staph aureus, that is a membrane depolarizing toxin. And then in Strep intermedius, there are toxins called TEL-B, which degrades NAD, and there is a toxin called TEL-C, which can alter the lipid 2 compound. And so again, to sort of summarize this, when the type 7 secretion system of an inhibitor cell produces these toxins, they can deliver them into a recipient cell, which can result in either growth inhibition or cell death. So we took a look at the OG1RF genome, and it turns out that OG1RF encodes a complete type 7 secretion system that is induced by this VPE25 phage. So on the top here, I'm showing a cartoon of the type 7 secretion system locus with the type 7 genes highlighted in magenta. And we see that we have many of the core genes that make up this core protein complex, such as ESAA, ESSC, and ESSB. And then this panel down here on the right is showing some qPCR that Anushala did to confirm these findings from the RNA-seq. So she took this OG1RF strain and added phage, and then did qPCR on some of the type 7 genes at 10 minutes, 20 minutes, and 40 minutes. And what you can see is that even as quickly as 10 minutes after infection with phage, some of these type 7 secretion system genes are turned on sort of 5 to 15-fold. So it seems to be a pretty quick, robust induction of this system. And it turns out that type 7 secretion systems are pretty widespread in Enterococcus, which is pretty interesting because they'd never been described in this organism before. So I know there are lots of lines on this phylogenetic tree. You don't need to be able to read all of them, but what I want to highlight is that the purple highlighted regions correspond to type 7 secretion system proteins in Enterococcus faecalis. These are also widespread in Enterococcus faecium, which I'm highlighting by these black bars here. And there was a paper a few years ago that kind of took all the species of Enterococcus and separated them into 4 Enterococcus species groups. And we were able to identify type 7 secretion systems in 3 of these 4 species groups, suggesting again that these systems are really widespread throughout these organisms. So what we wanted to know after this was whether the induction of the type 7 secretion system in OG1RF by adding phage led to inhibition of neighboring bacteria. So to test this, Anushala set up this really elegant experiment where she took Efacalis OG1RF, or a type 7 null derivative of this strain. So these can be targeted by phage. And she mixed them with a phage-resistant bystander strain in a test tube. And then she split this and either added phage or left this untreated with no phage. And then she quantified the surviving bystander bacteria in the test tube where she added phage relative to the test tube where there was no phage present. So if this ratio equals 1, then the bystander bacteria survived equally well in both conditions. And if the ratio is less than 1, then the bystander bacteria survived better without phage than with phage present. So if we look at the results of this experiment, if we look just at the black bars first, what we can see is that after 8 and 24 hours of co-culture with this phage, we see a 1 to 2 log reduction in viability of this bystander strain when co-cultured with OG1RF and with phage. But if we look at the type 7 null strain, we see that there's no difference in viability when the bystander is mixed with phage and without phage. And so we think that this indicates that this phage-induced type 7 secretion system is actually leading to inhibition of these bystander bacteria. And this is really exciting for many reasons, but this is the first demonstration that we're aware of phage regulating an antibacterial secretion system, which is kind of an unexpected finding. So we then wanted to know what the target range was of this phage-inducible type 7 secretion system. So Anushala did these same co-cultures with lots of different bacteria, and what she found was that when the type 7 secretion system was induced by phage, she could see inhibition of other gram-positive organisms, including Enterococcus faecium, Staph aureus, and Listeria. But interestingly, the system doesn't seem to be able to inhibit Streptococcus, and we're not really sure why at this point. She also found that there was no inhibition of gram-negative organisms, including E. coli, Salmonella, and Vibrio. And this isn't terribly surprising because studies of other secretion systems have found typically that the ones in gram-positives aren't able to inhibit gram-negatives. And so it seems that the system can inhibit a fairly wide range of all gram-positives, but it's not able to inhibit, every gram-positive that we throw at it. But this is really interesting, this ability to inhibit other organisms because, if we think back to the types of infections that Enterococcus can cause, we know that this can cause polymicrobial infections. And so I'm really curious in understanding how this ability to inhibit other bacteria or not inhibit other bacteria in the case of gram-negatives might contribute to some of those polymicrobial infections. So we next asked what toxins or effectors are produced by this type 7 secretion system that might be leading to this toxicity. So as I mentioned earlier, the toxins that are secreted by type 7 secretion systems typically have an N-terminal LXG-domain and a C-terminal toxin domain. When I looked in the OG1RF genome, what I found was that the system encodes 2 full-length LXG genes. And so what I have highlighted up here on the top are potential toxins in the type 7 secretion system locus. And then what I'm showing down here is that 2 of them, the gene numbers are 11109 and 11121. 2 of them encode gene products that have this LXG domain at the N-terminus of this protein, which suggests that these might be secreted toxins in this system. And interestingly, these other highlighted gene products here don't have the full LXG domain at the N-terminus, but they do share some sequence homology with other parts of these full-length genes. And this is a pretty common theme in bacterial secretion systems that have these sort of arrayed sets of toxins where they have smaller regions of homology that might facilitate homologous recombination. So you could, envision a genetic circumstance where through homologous recombination, you could maybe swap this toxin domain onto this full-length gene. And the way these toxin systems work is, so they're secreting toxins into neighboring cells, but they of course want to be able to protect themselves against self-intoxication by the production of these toxins. And so many toxins in these polymorphic toxin systems encode, they have an immunity gene found directly adjacent to the toxin gene. And so what this leads to when the proteins are translated is you have a toxin protein that can inhibit the growth of your target bacteria. And then you have an immunity protein, which is produced that physically binds and inactivates this toxin protein. So these are produced inside the cell making the toxins. And then this is what prevents self-intoxication. So I looked at the genes next to these putative toxins and found that there were some small genes that were candidates for these immunity genes. And that's what I've highlighted here with these dashed lines. So we asked whether any of these putative immunity genes could confer protection against toxicity from this type 7 secretion system. So we cloned several of these immunity genes onto plasmids and gave those plasmids to our target bacteria. And what we found by doing the co-cultures where we could induce the type 7 secretion system with phage was that one of these putative immunity genes, which is shown here in this dashed line in green in the yellow box, was able to offer more than a log of protection against killing by this type 7 secretion system. And so this toxin is a good candidate for what might actually be the delivered toxin, because if we express the putative immunity gene, we can block that inhibition. So we next asked whether disrupting this toxin prevented inhibition by this phage-induced type 7 system. So to do that, we did these same, or Nushla did these same co-culture experiments using a transposon mutant with an insertion inside this gene that we think is our putative toxin. And what she found is that this phenocopied the type 7 null mutant in that it was unable to kill the bystander bacteria used in this experiment. And in this case, it was a strain of Staph aureus. And we could complement this, or we could restore toxicity to Staph aureus by providing this toxin immunity pair on a plasmid, which sort of strengthens our conclusion that we've sort of identified the delivered toxin that's delivered into target cells by this type 7 secretion system and leads to growth inhibition. So unfortunately, we don't know what the actual toxic activity of this toxin is yet. So we've identified it, we know the immunity, and I've done a lot of bioinformatics looking at sort of the predicted protein structures and looking at sequence homology and whatnot. And we don't know what the actual activity is that's causing this toxicity to target cells. But if we look in sort of throughout Enterococcus type 7 secretion systems, we find that there's a really wide variety of toxins that are encoded by these systems. And so what I'm showing you here is a cartoon summarizing the diversity of toxins that are found in other Enterococcus type 7 secretion systems. And they're color coded by function. And so shown here in green, these are all different toxins that are predicted to be nucleases, so they could cut DNA or RNA. There's some that are predicted to have unknown function, and there's a handful of other domains. And so what I think this really highlights is that we've found just one type 7 secretion system in Enterococcus OG1RF, but kind of hiding throughout many other Enterococcus isolates are all of these really diverse toxin systems that can use all these different toxins to inhibit neighboring bacteria. So finally, we asked, in addition to phage, what else can induce this type 7 secretion system? And so Anushala grew OG1RF and treated it with many different antibiotic compounds, and then did qPCR to quantify gene expression changes or induction of the type 7 secretion system. And really interestingly, she found that expression of the system is upregulated in sub-MIC levels of daptomycin, of mitomycin C, of ciprofloxacin, and to a lesser extent, vancomycin. And this is really exciting because it shows that this is more of a general mechanism of induction of this system. It's not specific to phage. And I think this is really exciting, kind of connecting it back to infections and how we might treat those with antibiotics, and some of the sort of unforeseen consequences that could have because of induction of this system. I also think it's interesting to note that the transcriptional responses here were not all the same. So we saw that mitomycin C was able to really strongly induce the system, whereas vancomycin, barely had a twofold increase in gene expression. And lastly, we were interested in understanding whether this induction of the type 7 secretion system by sub-MIC levels of antibiotics could also lead to inhibition of other bacteria. And so Anushala, again, performed these co-culture experiments where instead of treating cells with phage, she treated the co-cultures of OG1RF and her target bacteria with sub-MIC levels of daptomycin. And what she found was that in liquid media, she could see a modest reduction of the target cells in a type 7 dependent manner. And because she didn't see inhibition of these target cells using the type 7 null deletion strain. And then on agar media, where the cells are much closer together and they're really packed onto a solid surface, she saw a larger degree of inhibition of these target cells in a type 7 manner. And so what this shows is that sub-inhibitory levels of antibiotics can induce this type 7 secretion system and lead to the inhibition of neighboring cells. So this brings me to my model of what we think is happening in this type 7 secretion system mediated inhibition of bystander bacteria. So we know that Enterococcus faecalis can sense phage and it can sense antibiotics through a mechanism that I didn't have time to talk about today. and that this leads to induction of expression of these type 7 secretion genes. And the core membrane components I've diagrammed here in this little cartoon membrane. We know that this leads to the secretion of this LXG-domain protein toxin, and we know that this can inhibit the growth of bystander bacteria unless they encode this specific immunity protein, which can block this toxic activity. So in conclusion, so what I've shown you today is that there are many models for using phage therapy to target persistent Enterococcus infections, but we have kind of a limited understanding of how phage and phage cocktails can work against Enterococcus biofilms in these different model systems. And quite excitingly, I've shown that some Enterococcus strains encode an antibacterial type 7 secretion system. And at least in our strain, this can be induced by phage and antibiotics. And so what I think this highlights is that much more work is needed to understand the interactions between phage and their target bacteria and how those interactions might impact surrounding bacteria, the surrounding microbiome. And some questions that I'm really interested in moving forward or sort of some of the big questions that I think we can think about as in the field is that, first, what are the activities of the toxins that are encoded by our system? We've identified one toxin, but we don't actually know what the molecular activity of that is, although we are working on it. Second, I'd like to know whether these type 7 loci in other Enterococcus species can be transferred to faecalis or between strains through horizontal gene transfer. That we know that Enterococcus is quite good at picking up or acquiring antibiotic resistance mechanisms or other mobile genetic elements. And it would be really interesting if we would see spread of these type 7 loci in the same manner, how that might affect a phage therapy or a response to antibiotics after acquisition of this element. I'm also interested in understanding whether this type 7 system can affect or predict colonization of the GI tract. Does having this system or lacking this system, does it affect the dynamics between other bacteria in the gut? And does it affect the clearing of polymicrobial infections by phage or by other antibiotics? And also in a related note, does this contribute to the ability of Enterococcus to really flourish and kind of overgrow in the GI tract after antibiotic treatment? And then moving forward, with developing some new phage therapy approaches, like I mentioned, I'm really interested in Enterococcus biofilms, and I'm really interested in understanding what approaches are best for clearing biofilm-associated infections, and whether the composition of the bacteria in the infection site, whether that matters, and also how the age of the biofilm, whether it's an early biofilm or whether it's a really well-established biofilm, affects susceptibility to phage therapy. And then to go along with that, I'm interested in sort of developing new in vitro models for studying some of these phage biofilm interactions. And with that, I'd like to thank the organizers for having me. It was really nice to be able to share this work. I'd like to acknowledge my postdoc advisor, Gary Dunny, at the University of Minnesota, and I'd especially like to thank my collaborators, Bracken and Nushla at UC Denver. We've had a really great, productive collaboration that I hope continues for a long time. And I am currently supported by the American Heart Association, so I'd like to thank them for funding. And with that, I'm happy to take any questions.
That was fascinating, Dr. Willett. Really cool work. Thank you so much for sharing your work with us. And I'm just going to remind folks to put in your questions in the Q&A format. So I'll start off. I was interested in this notion of this type of secretion system being present in Enterococci, but also other species, staph, and so forth. Forgive me if you've mentioned this, but is this sort of bystander effect also seen with staph and secretion systems in other organisms such as staph? And if so, what are you finding induces those, if not phage?
Yeah, that's a great question. Yes, so other organisms that carry these secretion systems can inhibit the growth of bystander bacteria through these same sort of classes of toxins. I believe in staph, it's known that one of the systems is inducible by lipids that are found in the host. And in some cases, in some strains of Staphylococcus aureus, the systems seem to be constitutively expressed. So they're on in lab conditions. I actually think that part of the reason nobody had studied this in Enterococcus before is because it's completely shut off in lab conditions, and it's, you know, expressed in these very sort of specific, after these very specific treatments. And so, yeah, it seems to be a range of things. You know, I think we found the first phage-inducible system. There are some that are constitutively active. There are some that are induced by host compounds. So kind of a range of things.
So, thanks for the wonderful talk, and to introduce myself, I'm Nick Chia. So sorry I didn't get to pop on beforehand, but you know, the one little bit of data that just came out in your talk that was so quick people might have missed it was the non-dose sensitivity. And so, do you think that's a general rule that the dose is not how were the doses tuned in those different experiments? Were they chosen kind of arbitrarily and they all worked, or were the doses tuned to what's effective, and different viruses have different dose needs?
Mm-hmm. Yeah, so do you mean the like the MOI of the phage that was chosen and yeah,
Right, because you had that example of like some that were, 10 to 3 even, or 10 to 4,
sure.
in terms of number of given to,
Mhm,
and this was a mouse model, I believe,
mhm,
and some were up at 10 to 12, and that's a pretty big difference. And so if that's arbitrary, it could indicate dose insensitivity.
mhm,
But if that was optimized, then it would seem like there's a huge dose range that we have to
mhm.
think about.
Yeah, that's a really interesting question. So in I know like in our specific cases, Breck and Anushala chose a specific MOI. We use a really low MOI actually. But I know, right, in some of those model systems, some of the studies did tune the dose to achieve a certain MOI based on the number of bacteria that they were sort of co-infecting. And I think some other studies have been guided by sort of what might be best practices in sort of phage therapy in terms of achieving kind of a standard dose. But I think it's kind of hard to say right now whether those studies, either the animal models or the ones that were done in vitro, right, if there's a dose dependency there or not.
So just to follow up before I give it back to Gina, it sounds like people were maybe or the plan was to calculate based on sort of a estimated number of bacteria and then having a target MOI, but it wasn't like people. I just want to make sure it's not like they tried different doses in that same animal to try to like what would happen if you half that dose, or what would happen if you know.
Sure, sure. It is no, I'm remembering back to those animal studies. I think most of those studies, yeah, chose one dose and went with it. Yeah, but so I'm not sure if in those specific animal models, if sort of doing a dose dependency curve, if you would find a threshold where the therapy is not effective.
Okay,
Yeah.
thank you.
So we have a question by Tao Masters. Great presentation. Did phage / antibiotic treatment that induced T7SS affect E. faecalis in terms of viability?
Yeah. Yes. So if the data that we're showing plots the bystander bacteria viability, but if you look at the E. faecalis viability, you are targeting it with a phage, and so you do see a reduction in Enterococcus viable counts because of that phage. For the antibiotic studies, everything was really carefully chosen to be sub-MIC levels. So for those studies, we weren't using a concentration of antibiotic that would kill the organism. But yeah, you do get lysis of, it's kind of the cyclical thing where the timing, it's a carefully controlled event where the cell that gets infected by phage must be able to induce the system and kill a neighboring cell before it succumbs to lysis because of the phage.
Hmm. So so as a follow up question, I was actually thinking about this. you had shown that the phage don't affect the microbiome, right? And I assume gut microbiome, and that's kind of interesting because you know, typically a lot of like enteric infections, you know, you would give antibiotics and that would disrupt the whole thing, and it's hard to say whether that's helping or hurting sometimes. Um, so uh, do you see high utility here, and what are some of the things you think we would need to check off our list? You know, some of the studies we need to check off our list to get there.
Yeah, that's a great question. I will point out, so a lot of studies that have been done with Enterococcus use the strain V583, which I mentioned, which is a vancomycin resistant strain, and that strain does not have a type 7 secretion system. And so, yeah, there's all sorts of really interesting genomics about, you know, strains that have it and strains that don't, and what that has sort of meant for the discovery of the system. But a lot of those animal models use V583 because it is a vancomycin resistant strain. You know, and vancomycin resistance is so critical for these organisms. And so I think it's possible. And, you know, we're working towards some of these experiments that if you repeated those same experiments with a faecalis strain that had a type 7 secretion system, you might actually see much greater changes to the microbiome. And so I absolutely think the presence of the system is something that could maybe predict how the microbiome will change after phage therapy. And in terms of, you know, how that might guide us in the future, I think this is, you know, one area where we could see a really great collaboration between sort of genomics and phage therapy was if we isolate, you know, something that's causing an infection and we sequence it and we find that it has a type 7 system that looks very similar to these ones that are induced by phage, we might be able to predict, okay, well, there are going to be some microbiome changes associated with phage therapy.
Yeah, no, I found that fascinating between the link between the functionality and the phage and then the phylogeny. I know you said we didn't have to be able to read that, but I like reading phylogenies. And so one of the things I found interesting there is it seems like it's indicative of a lot of horizontal gene transfer within that group. And and so it seems like it might be possible if you knew the host range, right? And you were tracking the genes and the horizontal gene transfers, it seems like they come together with the phage, right? They work they both are really horizontally gene transferred somehow.
Yeah, definitely. Yeah, so that phylogenetic tree was built on protein sequences for the conserved WXG100 effector that's present in all type 7 secretion systems. And yeah, the spread of that throughout Enterococcus is really fascinating. And the one striking thing that I very briefly mentioned was there's that one species group that includes Enterococcus casselflavius and gallinarium, where I could never find any examples of type 7 secretion systems in those organisms. So, you know, are we seeing horizontal spread throughout some parts of Enterococcus, but, you know, not into other species, and what might be restricting spread into those other species? I'm very interested in, you know, sort of pursuing the genomics of that and yeah, answering
Yeah,
some of those
no,
questions.
that's great. I'm always looking for excuses to look at horizontal gene
Yeah,
transfer.
oh, great.
I'm curious, maybe the flip side of that could be a potential reason why um, this secretion system tends to not target strep. Um, and you know, I don't know enough about the phylogeny to know, but morphologically, strep and enterococcus are very, you know, highly related. And I wonder if there's you know, some protection of organisms that are too similar to it itself.
Yeah.
Mhm.
Yeah, that's a really interesting idea. And right, we don't know why it doesn't target streptococcus. My leading theory right now is that maybe it's related to the cell wall. So one thing we don't know about these systems is whether or not they require a specific protein receptor on the surface of the bystander bacteria. So in some bacterial secretion systems, in the type 5 secretion systems, the cell that's being inhibited has to express a specific protein on the surface for the toxin to recognize to facilitate toxin delivery. But in the type 6 secretion systems, those form this sort of like needle that just punches through the cell wall, and that's how those toxins are delivered. And I don't think we quite know the delivery mechanism for type 7 secretion systems yet. So yeah, it would be fascinating whether, you know, the organisms are just too related, and so that's why there's no recognition there, or if there is a specific protein receptor, maybe that just isn't found in Streptococcus. Maybe there's something about the cell wall or the capsule that kind of precludes recognition. Yeah, that's a really, really interesting question.
Well, unfortunately, we are out of time, but that was wonderful and fascinating. Thank you so much again for sharing and teaching us today.
Yeah, thank you for having me.
Thank you. And then just a quick announcement, January 27th, we will have Cedric Lude of Dr. Rob Levine's lab talking to us from Brussels, Belgium. See you then.
Thank you.
Bye, everyone.